Students, welcome to Shubhra's Inkpot. Today in this video, we will discuss an activity to show chlorophyll is essential for photosynthesis. This activity is also known as iodine test or starch test. In this video, I will explain you about the steps as well as I will explain you the reason behind those steps in detail. This activity demonstrates that chlorophyll is essential for the process of photosynthesis. Step 1. Take a potted plant with variegated leaves. For example, money plant or croton. So students, what is the meaning of variegated leaves? It means leaves containing patches of various color. That is, it not only contains green color but contains other colors too. A variegated leaf is used in the experiment because some part of the leaf is green in color while some part is non-green. The green part of the leaf is green due to the presence of a pigment known as chlorophyll, and so it can perform photosynthesis. The non-green part does not have chlorophyll and hence it will not perform photosynthesis. Step 2. Keep the plant in a dark room for 3 days so that all the starch gets used up. The plant is then kept in the dark for 2 to 3 days so that all the stored food in the form of starch gets used up. There is no photosynthesis in the absence of sunlight and hence no further food formation occurs. Step 3. Now keep the plant in sunlight for about 6 hours. It is kept in sunlight for 6 hours to allow photosynthesis to occur. Carbohydrate or starch formation occurs due to photosynthesis only in green part of the leaves. Step 4. Pluck a leaf from the plant. Mark the green areas in it and trace them on a sheet of paper, tracing paper. The leaf containing starch is plucked and marked for green areas, which will help us to compare the results obtained further with the iodine test. Step 5. Dip the leaf in a boiling water for few minutes. After this, immerse it in a beaker or a test tube containing alcohol. Carefully place the above beaker or test tube in a water bath and heat till the alcohol begins to boil. The leaf is treated with alcohol because it acts as a bleaching agent and makes the leaf colorless. The setup can be easily explained through this diagram. Students, you can clearly see in this diagram how the test tube containing alcohol and leaf has been placed. Alcohol is never heated directly because it is highly flammable, so it is kept in a water bath and then heated with the help of a burner. Next step, that is step number 6. Now dip the leaf in a dilute solution of iodine for few minutes. Take out the leaf and rinse off the iodine solution. Observe the color of the leaf and compare this with the tracing done in the beginning. The green areas of the leaf turns blue-black because iodine reacts with the starch produced in the green areas due to photosynthesis, whereas the non-green areas remain colorless. In this diagram, you can clearly see the variegated leaf that in diagram A before the starch or iodine test and in diagram B after starch and iodine test. And you can clearly see that the portion which was green, photosynthesis happened there and it turned blue-black after the iodine test. So, this activity demonstrates that chlorophyll is essential for photosynthesis. I hope students, you understood this activity very well. You understood all the steps as well as you came to know about the reasons behind those steps, reasons behind performing those steps. And if this video helped you in understanding this topic very nicely, then do like, share and subscribe my channel. Thank you.